KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
Neet, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Education Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
In vegetative propagation and plants, so last and final one, modern method. So initially, so what are the methods we have seen now? Natural methods we have seen, later artificial methods we have seen, and now we are going to see modern method. What is a modern method and how we can produce so more number of plants within a short period of time, and what is a modern method now we will use. Now let us see one by one. Now don't forget, so vegetative propagation in plants, so definitely vegetative propagation of plants definitely takes place by first one natural methods, later artificial methods, later third one modern method. In a modern method point of view. So this particular information I have taken from our metric textbook. So it is also good. Let us study. So here even CBSE information also will be mixed. So thereby, so CBSE textbook and our metric state book and Alen Akash likewise. So there are different types of materials together. So KURINJI material is formed. Okay, so now let us see one by one. Now Professor F.C. Stewart. So in a modern method, first one is Professor F.C. Stewart of Cornell University showed that the mature phloem parenchyma cells removed from the carrot were placed in the suitable medium under controlled conditions. So what they have done, so scientist or the Professor Stewart, F.C. Stewart, and it belongs to the Cornell University. So what he was done means, so what he was did means the matured phloem parenchyma was removed from the carrot. Matured phloem parenchyma. Okay, matured phloem parenchyma was removed from the carrot and placed in the suitable medium, and it was placed in the suitable medium under controlled conditions. So means under controlled conditions means in laboratory conditions. Laboratory conditions are nothing but which in vitro conditions. In in vitro conditions. So this particular professor start to do experiment. So by taking off which one? Phloem parenchymatous cells. And when he was placed these phloem parenchymatous cells in a nutrient medium, suitable medium means which contains of nutrients. Okay, suitable medium under controlled conditions means in laboratory conditions, or laboratory conditions only we will be calling as in vitro conditions. Okay, so could be stimulated. So means when we will provide suitable conditions, when we will create optimum conditions for the growth of the plant, automatically what will be happen? Could be stimulated to start the dividing cells again and again to produce a new carrot plant. Point you understood here. So means here scientist or the Professor Stewart, so he was taken phloem parenchymatous cells, and these phloem parenchymatous cells he was placed in a suitable medium or placed on suitable medium, and he was provided all controlled conditions, means here all suitable conditions when he was provided. So automatically here we are observing the production of new plants. The development of new plants we are observing from the phloem parenchymatous cells. Next one. Okay, now these cells were described as totipotents. Means here, what are the cells? Once you see up in second line, phloem parenchymatous cells, scientist Stewart was taken for his experiments. No. So, what are the cells he was taken for the generation of a new carrot plant? So, mother cells means initially what are the cells we have been taken? No, all these cells now we can be taken those cells as totipotent cells. Why? Because these cells has the capability to give rise to a new young plant. Means in our studies we learned tissue culture, no, there. So one totipotency or totipotent, likewise we see. So means here the capability of the plant part or the capability of the plant cell to give rise or to give rise to entire plant is taken as totipotency. Explant means what is a plant we will be taking for our tissue culture techniques. No, that particular plant we will be taking as explant. EX apram plant. Explant. Okay, so means here this particular scientist Stewart took which one? Phloem parenchymatous cells and provided suitable conditions with controlled conditions, and later he was observed that one the producing of new carrot plant. So means here what are the cells he was taken? So for his experiments, all those cells we will be taking as a totipotent. Totipotent means, meaning one more time I have written here, the genetic ability of a plant cell to produce the entire plant under suitable conditions is said to be totipotency. Means here totipotency is nothing but genetic ability of a particular plant cell. That kind of genetic ability is there to produce the entire plant under suitable conditions. Means here, sir, that kind of ability for animal cells ki irka sir means no. So God has given only this kind of totipotency or genetic ability only to the plant cells, but this kind of genetic ability or this kind of genetic ability is not there with the animal cells. There just it is there or confined to the plant cell. So thereby here the genetic ability of a plant cell to produce the entire plant under suitable conditions we will be calling as totipotency. Am I right? Now let us move forward to discuss one by one. Now this characteristic feature, this characteristic feature of a cell utilized in horticulture, forestry and industries to propagate plants. So means when we have seen that the genetic ability of the plants, definitely we will use. So that kind of capability. So where and where or in which type of like industries or what are the aspects we will be developing. So because of this totipotency nature means, so here, so in horticulture techniques as well as forestry and in even in industries. So in different types of industries to propagate the plants within a short period of time. Am I right? Now the growth of the plant tissue in a special culture medium under suitable controlled conditions is known as tissue culture. Means here what is the information? So here the development of the plants or the growing of plants or the growth of the plant tissue in a special culture medium. In a special culture medium under suitable conditions means when we will produce the plants or when we will produce the plants in a suitable conditions and by providing of suitable conditions or the special culture medium, so definitely we can develop the plants. So that particular process we will be considering as tissue culture. So already I have given this explanation, so we know very well tissue culture there. So definitely the growth of the plant tissue where in a special culture medium under suitable controlled conditions. Suitable controlled conditions in vitro conditions or simply laboratory conditions. Okay, so that particular process we will be considering as tissue culture. Next one, micropropagation. So micropropagation is nothing but the regeneration of the whole plant from a single cell. So means here how many cells we will be taking, means even single cell also we can take for the producing of entire plant. So here by taking of particular single cell we can produce entire plant. So here what is a phenomenon we are following, means regeneration, means from this entire single cell we are producing entire plant so by the process of or by the method of regeneration. So means the regeneration of your whole plant from a single cell or a group of cells, tissue or small pieces of vegetative structures through tissue culture in large or huge quantities, so within a short period of time. So means here already we have learned this particular definition. So regeneration of a whole plant from a single cell or a tissue or which one, small pieces of vegetative structures. So here what is the method we will be following, means so tissue culture methods we will be following. So by following of tissue culture methods we are producing very huge quantities or the large quantities of the plants we are producing within a short period of time. Suppose if you are producing so many number of plants within a short period of time by following the tissue culture techniques, means so such kind of propagation we can be taken as micropropagation. Micropropagation. So thereby, sir, what is the use of this, or what are the uses of this tissue culture? So what are the uses, means here advantages I have written. So once let us discuss these 3 points now what I have written. Right, now plants which do not produce viable seeds or seeds that are difficult to germinate can be propagated by tissue culture. Suppose once you see the plants which do not produce viable plants. So in this particular, suppose if the plants are unable to produce viable plants, in this particular cases, so here we can use tissue culture techniques, or one more also is there. Suppose if the seeds that are difficult to germinate, suppose here seeds are there, all these seeds are difficult to germinate can be propagated by the tissue culture. So here first one is plants which do not produce viable seeds. Suppose when the plants are unable to produce viable seeds, in that particular case we can produce or we can use tissue culture techniques at the time. Suppose if you use these plants, here we cannot produce viable seeds. In that particular case, plants now we will be taking, and by following of tissue culture methods we can produce more number of plants. So at the time, no need to take or no need to produce seeds. And second case, once you see that second case point of view, seeds that are difficult to germinate, seeds are there. Second case, seeds are there, but these seeds are very difficult to germinate. So that kind of plants also very simply, very easily we can propagate by tissue culture methods. Next one, rare and endangered plants. Which one? Rare and endangered plants can be propagated. So rare and endangered plants, right, like vulnerable plants are going to be extincted. So some kind of plants also we will be propagated or we can propagate it. So later one more important point, disease free plants can be propagated. So by starting off or by doing of meristem culture. So this particular point already we have seen. So means here, even though entire plant is affected by the pathogens, so there also here we can see that one particular part which is not affected by the pathogens, that particular part name is meristem. So means that particular meristem will not be affected by any kind of pathogens. So thereby what is the part is being healthy, means in affected plant, their meristem is unaffected part. So thereby by using of meristem definitely we can produce disease free plants. This is Neet examination point of view important, try to save in your mind. So okay, so meristem culture definitely we can use definitely you can use in order to producing of disease free plants. disease free plants means free the disease varum adi illa. So what is the meaning? No, so definitely these particular plants will not consist of pathogens. Okay, disease free plants so definitely can be propagated by meristem culture. Now next one, disadvantages. What are the means here? One or two I have written. So the what are those? No, sterile conditions must be maintained, which added to the cost. So means here, so in order to obtaining of or in order to starting of tissue culture techniques, definitely, so what we have to create sterile conditions. So sterile conditions when we would like to maintain at that time, definitely, so that costs more. Why? Because suppose while starting of our tissue culture techniques, what we have to do, so all like what is apparatuses or what are the materials or what are the requirements we will use in a tissue culture techniques, all must be sterilized. For that, article must be there or different types of apparatus is required. So thereby, so definitely this we have to spend more amount so for this particular thing at the time, so that one we can be considered. And one more, if we would like to sterilize our hands, means at the time, some kind of sanitizers or some kind of at least some kind of alcohols we have to use for sanitize our hands. So likewise, definitely, so this particular case means here the maintenance of sterile conditions, so definitely it is maintained with added to the cost. So means definitely we have to spend somewhat amount for maintenance of sterile conditions. Next one more point, that is sometimes cows undergo genetical changes which are undesirable commercial or undesirable for commercial use. Ipo enna, sometimes, not all the times, sometimes callus undergoes which one genetical changes. So why? Because here these are all not natural conditions, definitely artificial conditions in artificial conditions. So we have to perform this entire explanation. In this particular case, what is a callus is there? A group of undifferentiated cells. A group of undifferentiated cells we will be considering as callus. So when this particular callus undergo genetical changes which are undesirable for commercial use. So suppose in any kind of changes comes in a callus, so here at the time, that particular plant we cannot use. Why? Because so why we are producing more number of plants within a short period of time, we would like to get more money, commercial usage. But there, suppose if any kind of genetical changes comes, means what are the desirable characters we have seen in these plants, so by changing or by mutations, now we could not see the presence of wanted characters in those plants or in those in those, thereby commercially we will not be benefited. My point you understood there. So thereby here the advantages are there as well as disadvantages also are there. Right, now up to here, so I can give you information regarding which one, in vegetative propagation in plants, natural methods, artificial methods, and one more is modern method. Next one, so this particular information exclusively so to be learned. So here along with other material, so I have given information which is there in our textbook. Right, now let us see one by one. Lifespan, the period is the maximum people ki theriyum, but even though we have to discuss, right, the period from the birth to natural death of an organism. Which one? Period, the period of time or the duration simply. But here they have given like that only, let us continue to learn like that. Right, the period from birth to natural death. Which one? Period, the period of from birth to natural death of an organism we will be considering as lifespan. Lifespan. Next one, lifespan of an organism or of organisms or not necessarily correlated with their sizes. Star mark, very very important. If lifespan is there, lifespan of the organisms are not necessarily correlated with their sizes. Means here, suppose chinna size is there, means lifespan within 1 or 2 minutes, and romba perusa irukano, romba lifespan is many years. Likewise, no chance. Means here the size of the particular organism is not correlated. So that's why here few examples I have given. So before start to give these kind of examples, no, one more time let us see the second statement's point of view. So this particular second line is important. Lifespan of an organism or of organisms are not necessarily correlated with their sizes. Am I right? Now the sizes of crows and parrots are not very different, yet their lifespans show a wide difference. Once you see there, what is the information? The sizes of crow and parrot. Once you see there, and the sizes of crow and parrot, if you see there, no, both are like more or less equal in size. That is all, no, very not different means more or less equal. Yet, even though their lifespans show a wide difference. Ipu morphologically seems to be same the sizes point of view, but even though sizes is a very similarities are there in a size point of view, but lifespan point of view definitely we can find out wide difference. So likewise, so thereby here the lifespan of that particular organism is not mainly depends on the size of the organism. So this is the best example we have seen. Even examples also we have to store in our mind. Next one, similarly, a mango tree. Mango tree, we know very well, mango tree has a shorter lifespan when compared to as compared to a peepal tree. So means here the comparison between the peepal tree and one more is mango tree. So in comparison of these two, no, so where we can find out very lesser lifespan. So mango tree has shorter lifespan when compared to the peepal tree. Likewise, so one more difference also we can find out. Next one, what about? So next one is whatever be the lifespan, death of every individual organism is a certainty. Means here whatever be the lifespan, death of every individual organism is a certainty. So definitely, so whatever may be that lifespan, definitely each and every organism must meet at what particular phenomenon or particular parameter, that is death. Means so thereby here what is the point mainly we can save in our mind means no individual is immortal. Very, very important. So even though we are poor, or even though we are rich, or even though we are in a middle, or like whatever it is, so definitely at one particular situation, definitely we have to meet which one? Death. So thereby here, so what is the main important point author giving us is no individual is immortal except. So now one important phenomenon, that is except single celled organisms. Means now here God has given some important phenomenon only to the single celled organisms. Means here even though we will be having more or million number of cells in our body, definitely we have to meet the death state. But here single celled organisms, so single celled organisms definitely will not meet the death. Definitely, why? Because so these single celled organisms are immortal. So thereby on our globe or on our earth, so what is there are million number of organisms are there, but only single celled like organisms only are immortal. So this point we try to save in your mind. Now let us start to move important concept, that is reproduction. Now reproduction point of view, it is defined as a biological process. So definitely this particular process we have to save in your mind. Now reproduction is a biological process. One more time, reproduction is a biological process. Once you try to say reproduction is a biological process, so in which an organism gives rise to young ones or offsprings similar to itself. So definitely, so reproduction means that one only we know very well. So definitely, suppose if one organism reproducing So by the process of reproduction means definitely offsprings formation takes place or a young one's formation takes place. These offsprings are definitely similar to that of parent. So that is a point here we have written. Next one, if offspring grow later, what is happening now? What is the offspring is produced from the parent? That particular offspring grow, mature and in turn produce new offsprings. So that's we know very well. So after but it takes some more time, that is only growth and maturity. So once the growth and maturity is completed, definitely that particular organism gives rise to, or it has the capability to give rise or gives rise to new offsprings. So thus there is a cycle of both growth and death. So definitely, so first of all birth process, later growth process and finally senescence or death process. So next one, reproduction enables, this is important point. Next one, reproduction enables continuity of a species generation after generation. So here definitely here our generations or their means so from our ancestors to parents to our generation to next generation to next fourth, likewise. So here generation after generation, so definitely here offsprings formation is taking place means everything because of the process of reproduction. So thereby now what is the point now I am going to give you is reproduction enables the continuity of a species. Continuity of species generation after generation. Idi don't forget important. Sariya. Next one, when offspring is produced by a single parent. So orange color information also important. Let us watch. That is when offspring is produced by single parent with or without involvement of gametes formation. Okay, so that particular process or by that particular reproduction we will be taking as asexual reproduction. So means here when your offspring produced by a single parent, this is important. When an offspring is produced by the single parent with or without involvement of gametes formation, this particular phenomenon we can be taken as asexual reproduction. And one more process also is there. When two opposite parents or two opposite sexes participate in a reproduction process and also involve fusion of male and female gametes. So that kind of a process we will be taking as sexual reproduction. Means here what is the main important difference we can be discussed between the asexual reproduction and sexual reproduction. Means in asexual reproduction single parent involvement is there and there is a chance or there is no chance to the gametes formation. So that kind of a process or that kind of reproduction we will be calling as asexual. But sexual point of view, definitely, so there are different types of parents must be required with opposite sexes. Definitely they must participate in the reproductive process, and so in that sexual reproduction process what is happening? The fusion of male and female gametes, the fusion of two different types of gametes are fused together and results in the formation of diploid zygote. So thereby that particular process we will be considering as sexual reproduction. I hope you understood this one. Right, now let us start to learn the asexual reproduction first. So here 4 lines are there. So these 4 lines also let us discuss, later let us stop. Right, sexual reproduction point of view. In this method, a single individual is capable of producing offspring. So here as we seen before, so in asexual reproduction only one single parent involvement or one parent involvement is there or one individual involvement is there. Even though one individual involvement is there, that particular individual also is capable of producing offsprings. As a result, what is happening? As a result, the offspring that are produced are not only identical to the one another, but also exact copies of the parent. Means here, what are the offsprings are produced from the single individual? So these offsprings in all terms and conditions, that is maybe genetically, that is maybe morphologically identical not only identical and exact copies of the parent. Not only identical, exact copies of the parent. So thereby how the parent is is there morphologically, same like offspring also will be there. So for that's why here one important definition we have to study, that is or one important term to be studied, that is clone. Already we have seen, but here Neet examination point of view also one more time I have to cover, that's why I have given here, right. Now what is the term clone is used to morphologically and genetically similar individuals. So thereby here the term is the term clone is used for what? So what are the offsprings or what are the individuals are similar like how? Morphologically and genetically. Suppose if the individuals are similar morphologically and genetically means, so such kind of individuals, so simply our subject point of view we can be termed it as clone. We can be taken as clone. So ఇది కొంచెం just information only I have started. So in the next video, so what are the types of asexual reproduction methods are there and what are the examples and entire information. So definitely I'll give you in next video. So try to watch this video and be happy and be safe at home. Okay ma, thank you.